Scientists have studied how SARS-CoV-2, the virus that caused COVID-19, use the human ACE2 receptor proteins as entry gates into cells. According to the researchers, understanding how the SARS-CoV-2 enters cells and comparing it to other coronaviruses is key to identifying treatments against COVID-19. According to the scientists, including those from Icahn School of Medicine at Mount Sinai in the U.S., identifying the features of the novel coronavirus which enable it to efficiently enter cells might lead to novel drug targets against COVID-19. In the new study, published in the journal PNAS, the scientists assessed how SARS-CoV-2, and the closely related SARS-CoV virus behind the 2002-03 pandemic, use the human ACE2 receptor proteins as entry gates into cells. By conducting lab tests to understand how synthetic versions of the novel coronavirus and SARS-CoV enter cells, the researchers identified key mechanisms by which SARS-CoV-2 evades the host immune system, and enters cells. According to the study, the SARS-CoV-2 RBD has higher ACE2 binding affinity than that of the 2002-03 SARS virus, supporting a more efficient cell entry in the new virus. However, the researchers said the ACE2 binding affinity of the entire SARS-CoV-2 spike is comparable to, or lower than that of SARS-CoV spike. Despite the potency of its RBDs binding to HACE2, the entire SARS-CoV-2 spike does not bind to HACE2 any more strongly than SARS-CoV spike does, the scientists wrote in the study. Based on this observation, they suggested that SARS-CoV-2 RBD, albeit more potent, is less exposed than SARS-CoV RBD. The hidden RBD in the novel coronavirus, according to the researchers, can evade the host immune system, potentially leading to insufficient immune responses and prolonged recovery time. The scientists also found that unlike SARS-CoV, cell entry of SARS-CoV-2 is preactivated by a molecule called proprotein convertase furin. To maintain its high infectivity while keeping its RBD less accessible, the study noted that the novel coronavirus relies on host protease activation. Host protease activation is a significant determinant of coronavirus infection, and a significant target for host immune surveillance and human intervention strategies, the researchers said. The high ACE2 binding affinity of the RBD, furin preactivation of the spike, and hidden RBD in the spike potentially allow SARS-CoV-2 to maintain efficient cell entry while evading immune surveillance, the researchers wrote in the study. According to the scientists, these features of the novel coronavirus may contribute to the wide spread of COVID-19. Successful intervention strategies must target both the potency of SARS-CoV-2 and its evasiveness, they concluded.
WaveMile